So we participated at the microbiology workshop, and the idea was to have another view of the diversity in soils of microorganisms in soil. So it won't be quantitative experiments and results, but just another view.
So we collected different soil samples around the institute. For example, some sand from the beach, some soil close to a tree and close to grass. And then we diluted it several times. And so we took a little bit of this diluted solution. We spread it on Petri dishes. And then after already one day, we were able to see some colonies growing on the Petri dishes. And so after 2 days and after 4 days, we counted the colonies on the Petri dishes for different diluting steps. And so we were able to count or to have an idea of the number of colonies per volume of soil. And so obviously we underestimated. But well, that's what's going on.
Okay, and then we had a look on the microscope. So that's a colony sand, a colony found in colonies found in the sand samples. So that we took from the Petri dishes and put under the microscope. And so what you can see is that the morphology, the shapes of the colony already can give an idea of the diversity of the bacteria, of the species that are here. And so we have some filamentous colony and some circular colony, for example, here. But the most present colonies were circular, actually. Just in the sand, we had these filamentous colonies.
I have a question already.
Go ahead.
Were they moving? Was this moving in the microscope, or were they stable?
No, the colony were not moving. No. But then after we, yes, no, we won't show movies. But. Okay, so we also used live dead dye coloration. So the principle is that the green dye enters every cell, whereas the red dye enters the damaged cell that. They are called dead, but they are not really dead. They are just damaged. And so the damaged cells appear like pink. But of course, dead are not real colors. We recolored them. But that's the principle. And so we could see that the health state of our colonies. And here you can see, for example, that most of the cells are healthy and some cells are damaged. Okay, so then we also enriched the soil samples with glucose, and we let it for 1 day, and then we looked directly the soil under the microscope, and here we used another dye. So we have the dye, the green dye from that I showed you before. So you can see the bacteria in green, and we also used the blue dye, the chalcofluor. Which is actually specific to fungi, and so they colored the fungi. And so here you can see an hyphae, and you can also see the internal structure of the hyphae. So here the septum. Okay. And yes, that's a very last experiment that we did very quickly. It's just with the chips. So you have like a lamella with pores and channels, and we injected our solution in the pores. And then after a few days, we could see that in the pores we had a high diversity. Different size of organisms, so protozoa, for example, and bacteria. Whereas in the channels, since the channels are very thin, we didn't have the biggest organisms. So here we just have the bacteria and also a diatom that was here. And so this is a method, this shows that this method is capable to discriminate organisms depending on their size.
petri dishes, we put like I don't remember, maybe 100 microliters of the diluted solutions. And so the solutions are diluted by like 1,000 or 10,000. So then we made the calculations back to have the number of colonies for 1 minute of diet.
Why does the red dye go into the damaged cells and not into the healthy cells?
I think it's not able to enter the cell when the membrane is intact. So the green dye is able to enter the cells even if the membrane is intact, whereas the red dye is not able to enter the cell if the membrane is intact. But I don't know the chemistry exactly.
As I understand you, Henry, you grow your colony by giving them some food. And I was wondering if what you observe is representative of what is in the soil, or if you could favor maybe one kind of bacteria that eats this very fast, and then observe something that is maybe not representative of what is the true diversity.
Yes, for sure we favor that. Bacteria that are capable to eat glucose. And anyway, in the petri dishes, even without enriching, but even without that,
There are some giants in the petri dishes.
yeah, and I mean not all the bacteria are as able to develop on petri dishes. So probably we have like 1% or less than 1% of the bacteria that we put that were able to grow on a petri dish. So of course that's a major issue.